Imagine trying to fix a really complex malfunctioning car engine, but instead of using, a diagnostic computer or a precision wrench to target a single stripped gear, you just you just pour highly corrosive acid over the entire vehicle and sort of hope the bad parts melt first.
Right, which sounds insane when you put it like that.
Exactly. But that brute force approach is essentially how we've treated cancer for the better part of a century. I mean, you find the lump, you cut it out, or you poison it systemically with chemotherapy, and you just kind of hope the collateral damage doesn't take out the host.
It is a devastatingly blunt methodology. But historically speaking, that reductionist approach made sense given the technological limitations we had.
Sure.
If you only have a microscope that lets you see a localized cluster of broken cells, you're going to treat it as a localized glitch. But that localized thinking is exactly why we've hit so many impenetrable walls in oncology today.
Because it's not just a glitch in a vacuum.
Exactly. A tumor isn't just a rogue cell dividing on its own. It's a highly organized, rapidly evolving, and fiercely resilient ecosystem.
And understanding that ecosystem, like mapping it, predicting it, and ultimately dismantling it, is exactly what we are focusing on today.
It's a massive shift in how we think about the disease.
It really is. So for those of you listening, whether you are, a medical student prepping for boards, an oncology fellow, or just an intensely curious self-taught learner, consider today's session a masterclass. We are doing a deep dive into the source material on this massive paradigm shift called systems biomedicine.
Yes, specifically based on the research in systems biomedicine approaches in cancer research.
Right. And our mission today is to comprehensively summarize these new approaches. We're going to translate the dense data into the actual mechanisms of action without, leaving the high-level science behind.
Which is crucial because systems biomedicine really forces us to zoom out. Instead of putting a single mutation under a microscope and just ignoring the rest of the body, it demands that we look at the network.
Right, the bigger picture.
Exactly. How does one tiny genetic modification ripple through the entire tumor microenvironment? I mean, how does it fundamentally alter the tumor's metabolism, its immune evasion tactics, its structural matrix?
So it's all connected.
Totally connected. If you want to defeat an adaptive ecosystem, you cannot just attack one node. You have to understand the entire web of communication.
Okay, let's unpack this. If we are going to treat an entire ecosystem, I guess the very first logistical hurdle is figuring out how to measure it comprehensively, right?
Right.
We need to identify the biological footprints the cancer leaves behind.
Yes, and that brings us to biomarkers.
Right. And searching for biological markers in oncology actually goes back further than most people realize.
Oh, significantly further. I mean, the very first cancer biomarker was identified in 1848.
Wait, 1848? Really?
Yeah, 1848. It was the Bence-Jones protein, which is the light chain of an immunoglobulin, and they found it in the urine of a patient with multiple myeloma. So the concept of looking for a diagnostic signature is nearly 2 centuries old. But systems biomedicine has completely redefined how we classify and utilize these markers. We don't just look for a single protein in the urine anymore.
We're looking at the whole board.
We categorize an entire biochemical landscape. We're looking at DNA markers like single nucleotide polymorphisms or SNPs and widespread epigenetic changes like CpG island methylation that silence tumor suppressor genes.
Plus, we're looking at circulating tumor DNA that gets shed into the bloodstream, RNA expression levels, massive arrays of protein biomarkers, altered glycans on the cell surface, all alongside high-resolution imaging and pathology.
Right.
But here's the problem with the old way of thinking, right? Relying on a single biomarker is it's like trying to understand a massive factory's entire production line by testing a single cup of water from the river downstream.
That's a great analogy.
Right. Because if the water is toxic, you know the factory is doing something bad, but you have zero idea how the machines are actually operating inside.
It's an excellent way to conceptualize it. A single downstream metric completely fails to capture the spatial and temporal heterogeneity of a tumor. And that realization is really the genesis of the omics revolution.
The omics revolution.
Yeah. Systems biomedicine relies on integrating multi-omics. So that's genomics, transcriptomics, proteomics, epigenomics, and metabolomics. We aren't testing a cup of water anymore. We are continuously monitoring the electrical grid, the supply chain, the assembly line, and the exhaust output of that factory simultaneously.
But the sheer volume of that data has to be staggering. I mean, you are talking about millions of variables per patient.
Well, it generates an overwhelming avalanche of data. And that is exactly why systems biology is inextricably linked with computational biology.
Right, because a human brain can't track all that.
We can't. So we rely on massive cataloging databases like TCGA, the Cancer Genome Atlas, to warehouse this multi-omics data. And we use high-throughput technologies to generate it. Take mass spectrometry and proteomics, for instance.
Right, because we aren't just running simple assays anymore. With mass spectrometry, we are essentially taking a tissue sample, vaporizing it, ionizing the constituent molecules, and then accelerating them through a magnetic field, right?
To measure their exact mass-to-charge ratio.
Precisely. It gives us a hyper-accurate, high-resolution map of every single protein actively being expressed in that tumor at that exact moment.
Which is incredible.
What's fascinating here is how we apply it. For example, utilizing multi-omics data, researchers isolated the expression patterns of a specific protein called PYCR1.
And that's an enzyme involved in protein biosynthesis, right?
Exactly. And by tracking it through the network, they found that PYCR1 expression isn't just a byproduct of tumor growth. Its upregulation can actually accurately predict if a breast cancer tumor will develop resistance to a specific pharmacological intervention.
Oh, wow. So we move from reacting to a growing lump to actually predicting the ecosystem's defensive maneuvers before they even happen?
That's the power of systems biomedicine. So if multi-omics gives us the comprehensive map of the river basin, we can trace those signals back up to the factory floor to see how the cancer is actually keeping the lights on. And that is where the ecosystem goes completely rogue. We need to talk about the Warburg effect.
Yes. The Warburg effect is the foundational mechanism of how cancer rewires its internal circuitry.
Okay, break that down for us.
So in a healthy cell, under normal aerobic conditions, metabolism relies on oxidative phosphorylation in the mitochondria to generate ATP. It's highly efficient. Cells really only default to glycolysis, which is a much less efficient anaerobic process when oxygen is severely depleted.
Right, like when you're sprinting and your muscles burn.
Exactly. But cancer cells systematically hijack this network. They heavily upregulate glycolysis even when oxygen is perfectly abundant.
And on the surface, that sounds like a terrible evolutionary strategy. Like, why would a hyper-aggressive, rapidly dividing cell choose a deliberately inefficient engine to generate its energy?
Well, it only looks inefficient if you are solely focused on the ATP output. But cancer cells aren't just trying to survive. Their primary imperative is to multiply.
To build more cells.
Exactly. To undergo rapid mitosis, a cell needs a massive amount of physical biomass lipids for new cell membranes, amino acids for proteins, nucleotides for DNA replication.
Oh, I see.
By shunting glucose through aerobic glycolysis, the cancer cell deliberately creates a bottleneck. It forces the accumulation of glycolytic intermediates, these carbon skeletons, which are then diverted into side pathways like the pentose phosphate pathway to physically build those raw materials.
So it's not about energy, it's about building blocks.
It is a brilliant supply chain hijacking.
And it is driven by oncogenic mutations that force the accelerator to the floor, right? We are looking at mutations in pathways like RAS, EGFR, MYC, and BRF. They basically physically lock the cellular signaling in a state of constant resource hoarding.
Yes, and the clinical reality of this metabolic rewiring is terrifyingly fast.
Take non-small cell lung cancer, or NSCLC, for example. Because this rogue engine provides limitless building blocks, the tumor aggressively invades the mediastinum. By the time a patient reaches stage 3 or 4, the cancer has often infiltrated the lymph nodes above the collarbone, the pericardium around the heart, the major blood vessels.
It's devastating.
The ecosystem has entangled itself so deeply into the patient's vital infrastructure that surgical resection is clinically impossible.
And you know, that metabolic rewiring is really just one mechanism of evasion. Systems biomedicine also looks at how the fundamental instruction manuals are altered through fusion transcripts.
Right, the fusion genes.
Yeah. This happens via massive chromosomal rearrangements, translocations, inversions, or interstitial deletions.
Right. So if we visualize the genome as a set of mechanical blueprints, a fusion transcript isn't just a typo on page 4. It is taking the front half of the blueprint for a high-speed blender, tearing it in half, and physically stapling it to the back half of the blueprint for a gas-powered lawnmower.
That is a terrifying machine.
Right. The cell's machinery reads that new chimeric blueprint and builds an incredibly dangerous, highly erratic protein that shouldn't exist in nature.
That staple blueprint analogy is biologically very accurate. In a fusion gene, you frequently see the regulatory promoter region, the on switch, from a highly active gene seamlessly fused to the coding sequence of kinase or transcription factor from a different gene.
So it's just running constantly.
The result is a chimera protein that drives aggressive, unchecked proliferation, but but is constantly stuck in the on position.
But from a targeted therapy perspective, that is actually a massive vulnerability, isn't it? Because that stapled together blender lawnmower protein only exists in the cancer cell. The healthy tissue is reading normal blueprints.
Yes, that is the silver lining of structural systems biology. Understanding the exact biogenesis of these chimeric RNAs provides us with an exclusive biological target.
Which is huge.
It really is. It's accelerating the development of highly specific RNA-based therapies, like siRNA or antisense oligonucleotides that are designed to only bind to and degrade those specific mutated fusion transcripts, leaving the healthy cellular network completely untouched.
Which perfectly transitions us to the next massive shift in this field. Traditional chemotherapy, as we established earlier, is the grenade under the hood. It causes severe systemic collateral damage because it attacks any rapidly dividing cell hair follicles, intestinal lining, bone marrow.
It doesn't discriminate.
Right. So we need tools that fit the exact biochemical shape of these rogue metabolic pathways and chimeric proteins. And ironically, to find the most advanced, highly specific molecular keys, systems biomedicine is turning back to the most ancient chemical warfare on the planet: medicinal plants.
It is a profound realization, honestly. I mean, for hundreds of millions of years, plants have been locked in an evolutionary arms race.
Yeah.
Because they cannot physically run from predators or pathogens, their only defense mechanism is chemical.
They have to stand their ground.
Exactly. They have evolved an astonishing library of secondary metabolites, alkaloids, flavonoids, terpenes, designed to target specific biological pathways in herbivores and microbes.
And we can use those.
Yes. Systems biomedicine isn't looking at these plants as holistic teas. It is isolating these exact compounds to use as precision ammunition against the cancer network.
Let's examine the mechanisms of action for a few of these because the sources get really specific. The material spends significant time on Peganum harmala, widely known as Syrian rue, and specifically an alkaloid compound it produces called harmine.
Harmine is a fascinating multi-target compound. In the context of breast cancer, it directly addresses one of our biggest clinical hurdles, which is multi-drug resistance.
How does it do that?
Well, cancer cells often survive chemotherapy by massively upregulating transmembrane proteins called ATP-binding cassette transporters, specifically the BCRP efflux pump.
Okay.
The cell essentially uses these pumps to physically bail the chemotherapy drugs back out into the extracellular space before they can do damage.
Like bailing water out of a sinking boat.
Precisely. Harmine binds to and specifically inhibits the BCRP pump, trapping the chemotherapy inside the cancer cell.
It essentially blocks the exhaust pipe, and in pediatric neuroblastoma, it operates on a different axis entirely, right?
Yes, it triggers apoptosis by cleaving PRP, but it is selectively devastating to neuroblastoma cells that exhibit an amplification of the MYCN gene.
Now, MYCN amplification normally drives aggressive proliferation and correlates with a really grim prognosis. But harmine exploits that specific network state. And in leukemia, it actively suppresses DNA methyltransferase 1, which stops the cancer from epigenetically silencing the body's natural tumor suppressor genes.
It's incredibly versatile. And we should address a critical distinction here because I know you noted the toxicity profile of the crude plant in your notes before we started.
Yeah, I was looking at the botanical data, and crude Peganum harmala is highly toxic. I mean, ingestion can cause severe hepatotoxicity, liver degeneration, paralysis, and severe gastrointestinal distress. It seems counterintuitive to use a potent toxin to replace chemotherapy.
I get that. And that is exactly where systems biomedicine diverges from traditional pharmacognosy. If a patient consumes a crude extract or a tea, they are taking in hundreds of distinct, uncharacterized compounds at completely unregulated concentrations.
Right, it's a scattergun approach.
Of course, the systemic toxicity is severe in that case. But systems biology operates on extreme isolation and precise molecular targeting. We're extracting solely the harmine alkaloid, understanding its exact pharmacokinetic profile, and engineering the dosing so it only reaches the binding threshold for the BCRP pump or the DNA methyltransferase enzyme without overwhelming the hepatic system.
Precision over volume. The molecule is just a tool. The dosage and targeting dictate if it's a poison or a cure.
Exactly.
Now, another fascinating mechanism comes from Melissa officinalis, which is common lemon balm. Like, you can grow it on a windowsill. But its clinical application isn't just directly attacking the tumor. It acts as a molecular shield for the patient.
Correct. The key active compound here is rosmarinic acid. While it does show anti-proliferative effects against colon cancer pathways, its role as a cardioprotectant during aggressive chemotherapy regimens is incredibly promising.
Let's look at the doxorubicin mechanism. Doxorubicin is a foundational anthracycline chemotherapy used for breast cancer. It works by causing double-strand DNA breaks via topoisomerase II. But the major clinical limitation is severe cardiotoxicity.
It's a huge problem for patients.
Right. Doxorubicin generates massive amounts of reactive oxygen species in the heart tissue. This oxidative stress acts as a tripwire, activating the NF-kappa B transcription factor, which basically sounds the cellular alarm and floods the cardiac tissue with severe inflammation, ultimately causing heart failure.
Exactly. And the extract from Melissa officinalis intercepts that specific cascade. By introducing rosmarinic acid, you physically suppress the activation of those inflammatory transcription
It
factors.
just shuts them down.
It downregulates NF-kappa B, TNF-alpha, and COX-2. It doesn't repair the heart after the fact. It literally cuts the wire to the NF-kappa B alarm system so the inflammatory floodgates never open.
That's amazing.
It really is. This allows the oncologist to maintain an effective dose of doxorubicin without destroying the patient's cardiac network.
It is brilliant network manipulation. You also see this with Quercus infectoria, specifically the galls. Galls are essentially plant scar tissue. They are irregular, hyperdense growths triggered by the biological stress of an insect depositing eggs or enzymes into the plant.
Right, it's a defense mechanism.
The plant's localized immune response generates a massive concentration of defensive tannins and phenolic compounds.
Primarily ellagic acid and gallic acid. And when you introduce these compounds into the cancer network, they act as broad-spectrum signal jammers.
Signal jammers, I like that.
Yeah, they have been shown to directly inhibit EGFR,
Wow.
which, as we discussed earlier, is one of the primary accelerators of the Warburg effect. But they also interrupt the AKT and JAK-STAT signaling pathways.
And those pathways are the communication lines the cancer uses to survive stress and initiate migration. So, if you block AKT and JAK-STAT, you effectively halt metastasis. The cell loses its ability to navigate and invade neighboring tissue.
Exactly.
Furthermore, it prevents the tumor from establishing its immunosuppressive microenvironment, basically unmasking it so the patient's own T cells can recognize and destroy it.
Now, here's where it gets really interesting. We have established the diagnostic map with multiomics. We understand the physical machinery of the Warburg effect and fusion genes, and we have identified these incredibly complex ancient molecular keys from nature.
We have the pieces.
Yes. But the final hurdle of systems biomedicine is, how do we actually put the key into the lock? How do we take a plant tannin and reliably engineer it into a pharmaceutical drug?
We move out of the wet lab and into the digital laboratory. Because the multiomics data we generate, the genomic mutations, the protein folding data, the metabolic cascades, it all results in billions of interconnected data points.
It's too much.
Right. A human brain, or even a room full of researchers, physically cannot calculate the correlations. So, systems biomedicine relies heavily on artificial intelligence and machine learning.
AI is the ultimate pattern recognition engine. It can ingest the multi-omics profile of a specific patient's tumor and accurately calculate survival probabilities, metastasis risks, and predict the exact efficacy of a targeted compound based on hidden variables a human would never catch.
But AI is only as good as the physical data you feed it. To design a molecular key, the AI needs to know the exact atomic shape of the lock.
And that is where structural systems biology comes into play, utilizing techniques like X-ray crystallography. This is the engine behind fragment-based drug discovery, or FBDD.
Okay, walk us through that.
So X-ray crystallography is a masterpiece of biophysics. You take the target cancer protein, say the BCRP efflux pump we discussed,
Right.
and you force it into a purified, crystallized lattice structure. Then you bombard that crystal with high-energy X-rays.
Sounds intense.
It is. The X-ray beams hit the electrons of the protein and scatter, creating a complex diffraction pattern. By mathematically analyzing that pattern, we can construct an electron density map, which gives us the precise three-dimensional atomic structure of the protein.
So you can actually see it.
We can literally see the grooves, the pockets, and the binding sites at an atomic resolution.
And once that 3D atomic map is rendered in the computer, we enter the molecular docking phase. The research highlights QSR quantitative structure-activity relationship.
Yes.
We aren't mixing chemicals in a beaker and hoping they bind anymore. We are computationally simulating the van der Waals forces, the hydrogen bonds, the exact thermodynamic interaction of a drug molecule docking into the cancer protein.
To illustrate how precise this is, the literature points to the mapping of the FS2 toxin from the black mamba snake.
Oh, wow.
Yeah, By using molecular dynamic simulations, structural biologists mapped the exact subatomic mechanism of how that specific venom peptide inserts itself into human calcium channels to trigger rapid cardiac arrest.
That is wild.
In systems biomedicine, we apply that exact same computational physics to oncology.
We take the 3D digital rendering of the harmine molecule from Syrian rue, and we digitally simulate sliding it into the binding pocket of the BCRP resistance protein. The algorithms calculate the exact binding affinity.
And if the natural plant molecule doesn't fit perfectly, the AI can digitally tweak the carbon structure. It can add a hydroxyl group here or remove a methyl group there, artificially optimizing the compound before a chemist ever physically synthesizes it in a laboratory.
That is the ultimate synthesis of systems biomedicine. We combine structural biology mapping the precise 3D atomic shapes with systems biology understanding the massive cascading network of how those shapes interact. It completely removes the guesswork from drug discovery.
It really does. We map the network, find the compromised node, and computationally engineer the exact atomic wrench required to shut it down.
Okay, let's take a breath and synthesize everything we just covered. We explored the paradigm shift of treating cancer not as a localized error, but as an adaptive ecosystem. We moved past single biomarkers into the high-resolution mapping of multi-omics.
Right.
We detailed how the Warburg effect physically hijacks the metabolic supply chain to build biomass, and how fusion transcripts stitch together rogue blueprints. Then, we investigated how natural compounds like harmine and ellagic acid serve as precise signal jammers. And finally, we looked at how AI and X-ray crystallography allow us to computationally dock these molecules with atomic precision.
It is a phenomenal amount of complex biology to process. And because we promise a masterclass for our medical learners and intensely curious listeners, let's do a quick structural review exercise to lock in the retention.
Let's do it.
Think back to our discussion on natural compounds acting as cardioprotective shields. Based on the pathways we covered, if you were treating a patient suffering from severe oxidative stress and heart toxicity due to the anthracycline doxorubicin, which medicinal plant from today's deep dive offers that protective buffer. And critically, what specific inflammatory transcription factors does its active compound downregulate to prevent heart failure?
Take a second to trace the pathway for yourselves at home. Got it. The answer is Melissa officinalis, or lemon balm. The rosmarinic acid physically cuts the wire on the alarm system, preventing the activation of the inflammatory factors NF-kappa B, TNF-alpha, and COX-2.
If you successfully track that mechanism, you possess a highly functional understanding of modern systems biomedicine.
So what does this all mean as we look to the future?
I think it opens the door to an incredibly provocative future regarding synthetic biology. We have spent the last hour discussing how we isolate natural compounds from plants that evolved over hundreds of millions of years and how AI optimizes them to fight cancer.
Right.
But as our molecular docking algorithms and quantum computing capabilities become flawless at simulating these atomic interactions, we are rapidly approaching a threshold where we won't need to harvest plants at all.
Wait, you are talking about AI moving from optimizing natural compounds to engaging in entirely digital evolution.
Yes.
Using these structural networks to design completely synthetic, non-natural plant molecules from scratch compounds that have never existed on Earth, grown entirely inside a digital greenhouse, specifically engineered to dismantle a single patient's unique tumor network.
Exactly. It shifts medicine from discovery to pure biological architecture.
That is an astonishing thought to leave on. Thank you all for joining us on this deep dive. Keep questioning the network, keep learning, and we will see you next time.